Parkinson's disease is such a complex disease and one of the most prevalent neurodegenerative disorder, and sometimes 2D models fail to convey that complexity of the disease. My name is Inês Silva. I'm a second-year PhD student. I'm working at the Molecular Nutrition and Health Lab at Nova Institute for Medical Systems Biology. And my research topic is actually constructing an NVU on a chip, but using the BCOO2 from Dynamic 42. And with that model, try to mimic the Parkinson-like phenotype, and thereafter tests our phenolic metabolites that we have in-house on, well, the disease pathology. Parkinson's disease is such a complex disease and one of the most prevalent neurodegenerative disorder, and sometimes 2D models fail to convey that complexity of the disease. And so I'm trying to implement a more physiologically relevant model that bypasses animal studies and, well, study the crosstalk between the brain cells and the BBB, and that's it. So we have our chip, and we have two channels, the BBB channel and the brain channel. On the BBB side, on the blood side, as we call it, we have the brain microvascular endothelial cells, and on the top channel we have dopaminergic light neurons, astrocytes, and microglia. Then we want to establish the PD-like phenotype by introducing two of the main, or mimicking two of the main hallmarks of the Parkinson's disease, a neurotoxicant and a pro-inflammatory mixture. And then when we have that model set up, we want to test our phenolic metabolites that we know are highly present in circulation as a preventive approach to Parkinson's disease. So my studies so far are actually implementing a model. So the results I have are on that area per se. So we firstly wanted to implement the cells. There are 3 cell lines, so they don't really love to be next to each other, so we had to manage a way to put them all together, and we finally did that, and we could reach the culture up to, actually this is not correct because we managed to do it for 14 days. And then we measured, we tried to implement our PD-like phenotype by introducing the things I said, the neurotoxic and the pro-inflammatory mixture, and we have, and we chose this stimulus because of these results, the cell viability and also the release of pro-inflammatory cytokines. They were the most promising results. And then we tried to apply the polyphenol metabolites on the system. And we had promising results in terms of cell viability, once again, and the release of pro-inflammatory cytokines. There was a reduction, which was really promising. We are very excited. We know it's going to be a challenge, but we are going to use the chip that allows us to have brain spheroids using the same cell lines I mentioned, but in a different setup, more three-dimensional, and it's pretty exciting. I think reading is very important. Do your research and go to this type of conferences and mingle with other scientists, and it's really great. You learn a lot. Then, and in that sense, you should collaborate. Collaborating is a great way of pushing your research forward. And lastly, but most importantly, be patient. Because this is hard, we know. But you'll get through it, you'll do it. One way or another, you'll manage, believe me.